Welcome. I'm Katya Brossart, Medical Science Liaison for Natera Organ Health, and today I'm presenting this review article entitled HNF1B-associated renal and extra-renal disease, an expanding clinical spectrum. This was authored by Clissold et al. And published in Nature Reviews Nephrology in February of 2015. So this is not a newer article, but provides a really good in-depth discussion of this important topic. I encourage you to go back and read this. This is intended to be kind of an introduction and a high-level overview. Disease associated with hepatocyte nuclear factor 1 beta, or the HNF1B gene, was first reported in 1997, and this was actually something of an incidental finding. Researchers were looking at the association between HNF1A and maturity onset diabetes of the young, or MODY, and discovered the important association of HNF1B with developmental kidney disease. And the HNF1B gene has since been recognized as the most common contributor to monogenic developmental kidney disease. It is associated with ARCAD syndrome, or renal cysts and diabetes, and is very much a multi-system disorder with a high degree of phenotypic variability. Looking at HNF1B molecular genetics, we see that this is located on chromosome 17 at position Q12. It is involved in regulation of gene expression. It makes up the gene product makes up a protein dimer, often in combination with HNF1 alpha. And pathogenic variants in HNF1B can range from base substitutions to small insertions and deletions, and very commonly whole gene deletions, which I'll talk about next. There's not strong evidence for a correlation between the type and position of variant and the clinical features, and we do see strong intrafamilial variability with this gene, and this suggests that there are other genetic and environmental modifiers at play that are not yet well understood. As I mentioned, deletion of the HNF1B gene is a common mechanism by which the related disease occurs. And very commonly, this is due to a larger 1.4 megabase heterozygous recurrent deletion at 17q12 encompassing HNF1B and contiguous genes. The authors note that deletion of HNF1B is present in about 59% of affected adults and 56% of affected children and fetuses. They also noted that about half of those HNF1B deletions are de novo, so occurring for the first time in that affected individual, as opposed to being inherited from an affected parent. More recent studies indicate that it is likely that the de novo rate for HNF1B deletions is even higher than that. The deletion syndrome is characterized by structural or functional kidney and urinary tract anomalies, maturity onset diabetes of the young type 5, and lastly, neurodevelopmental and neuropsychiatric disorders. Those do tend to be related to the recurrent deletion syndrome as opposed to effects of a defect in HNF1B itself. The authors also discuss the role of HNF1B in kidney development, noting that renal effects have been replicated in model organisms, in zebrafish, frog, mouse studies in particular suggesting an important role of HNF1B in early urogenital development. Renal-specific inactivation of HNF1B in mice has been shown to result in polycystic kidneys and reduced expression of key genes UMOD, PKHD1, and PKD2. HNF1B has been shown to be involved in several stages of nephrogenesis, and defects in HNF1B have been found in about 20% of cases of CACET, or congenital anomalies of the kidney and urinary tract. It results in widely variable renal phenotypes, cystic disease and cystic dysplasia being the most common effects, those being seen in about 73% of children and adults. Cysts are typically with HNF1B small and arising from the renal cortex, and typically do not progress in number. And unilateral renal agenesis is another common feature. There are certain electrolyte abnormalities also reported more commonly in individuals with HNF1B related disease. Hypomagnesemia is a common feature. Hyperuricemia is less common, but also reported along with early onset gout. Renal function ranges a great deal from normal to kidney failure, and in adulthood, slowly progressive decline in renal function has been described in affected individuals. There is still less known about the decline or progression in the pediatric population. For patients who eventually are transplanted due to kidney failure, there is an increased risk of new onset diabetes after transplant. And so these are patients who may benefit from a pancreas transplant as well, and also consideration of an immunosuppression regimen that would avoid agents that are likely to increase the occurrence of diabetes after transplant. As we can see in this illustration, there is a wide range of renal and extrarenal characteristics of HNF1B associated disease. So starting at the top left, we see those neurological features that are primarily associated with the deletion on chromosome 17 at Q12. So likely the effects of those contiguous genes that are deleted, the autism spectrum disorders, and cognitive impairment. Below that, we see the effects on the pancreas, early onset diabetes, pancreatic hypoplasia being fairly common, and below that, a range of genital tract malformation. So something that's just important to note if taking family history. Also, we see abnormal liver function is a common feature, particularly an asymptomatic rise in levels of liver enzymes. So a common finding here, with neonatal cholestasis being a more rare finding. And again, we see developmental kidney disease. So a wide range of effects, bilateral hyperechogenic kidneys on prenatal ultrasonography being a fairly common presentation, the renal cysts, unilateral renal agenesis, and renal hypoplasia being common as well. Other things that may be seen are horseshoe kidney, duplex kidneys, collecting system abnormalities, and bilateral hydronephrosis. Also, again, the electrolyte disorders of hypomagnesemia and hyperuricemia with early onset gout. The authors also discuss areas of potential future research. They note that it would be helpful to establish the prevalence of HNF1B gene anomalies in the general population, noting that this is very likely an underdiagnosed condition due to the wide phenotypic variability and the high frequency of de novo deletions. They also make a note that stratifying large patient cohorts by phenotype may be helpful in identifying determinants of that phenotypic variability. In summary, HNF1B related disease is the most frequent monogenic cause of developmental kidney disease. It is a multi-system disorder with quite marked clinical heterogeneity, often occurring in the absence of a positive family history. To end with a quote from the authors, HNF1B genetic testing should be considered for all patients with developmental renal disease, particularly if renal cysts or hyperechogenicity are detected or other extra-renal clinical features are present, end quote. I hope you've enjoyed this presentation of the Clissold et al. Review article, and I encourage you to read the article in its entirety if you're interested in learning more about HNF1B related disease.